Okay.
Okay,
So,
we are live.
perfect. So, hello everyone, and welcome to the virtual seminars on complexity hosted by the Center for Complexity and Biosystems of the University of Milan. I'm glad to welcome Dr. Annie Kaplanian, our today's guest speaker. I would like to shortly introduce Dr. Kaplanian. So she's a biologist specialized as a neurophysiologist on ex vivo electrophysiology, and currently she's employed in the Department of Neuroscience and Rare Diseases in Hoffmann-La Roche in Switzerland. Her research is focused on models of genetic epilepsies and finding new potential therapeutic targets using electrophysiological tools. And today she will talk to us about the physiology of sleep, its interplay with epilepsy.
Thank you, Mariana. Thank you very much. Hello, everyone. Thank you for the invitation. It's a great honor for me. I'm very happy to be here with you. So today, actually, I will try to keep it simple because it's a very complex topic, sleep, and we will go through the physiology of sleep, and in the end, I will try to give you some evidence on how actually brain circuits during sleep we can actually say that have similar mechanisms, and in the end, they have an interplay with epileptic seizures. So first, I will give you a first impression of a general impression of sleep physiology. We will try to understand how the brain decides to go to sleep and when it decides to wake up. We will talk a bit about the sleep stages, the sleep architecture, and the brain rhythms, and we will end up talking about slow oscillations, which is the major topic for us today. Second, I will show you some results, work that is part of my PhD work and is ex vivo electrophysiological work on slow oscillations, and data on how this rhythm transits into epilepsy. And we will make an overview on that. So let's start. Us humans, and not only humans, spend 1/3 of their lives sleeping. This is enough to say that sleep is very important for our lives, but yet we know very few things about it, its role and its physiological role, and what is the impact of sleep in our lives and in our brains. So let's try to understand some more things. First of all, in order to understand circuits and brain networks, we need to see every neuron, every neuronal cell, as an oscillator. So a source that can actually produce electrical signals. The neurons that are producing electrical signals, they are oscillating individually and all together. So they communicate by electrical signals, creating networks, and this network activity is in the end what we see, what we record in the EEG. So I will show you some network activity, these kind of signals which characterize different brain states during sleep. And I want you to keep in mind that every kind of signal like this comes from the network activity from the neuronal population. So actually during sleep, what brain does is that subcortical regions of the brain communicate with the cortex, and actually the signals we see in EEG are mainly the cortical activity. So sleep is actually, we have a hypothesis about sleep, is let's say a competition between two processes, or not exactly a competition, but working together as well, that balance wakefulness and sleep. So it's the process S and the process C. The process S is all the homeostatic mechanisms that tell the brain, okay, now you are alert, now you wake up, or now you shut down and you go to sleep. And the process C, which is the circadian rhythmicity of the brain, is an internal clock that we have in our organism, and modulates the brain activity, but not only the brain activity. During a 24-hour clock. So actually, the circadian rhythmicity is very important for us to sleep, to go to sleep, but without that, we would sleep again. It's not that if we move out the circadian rhythmicity, we cannot sleep. We would still sleep, but then it would be a random sleep around the day. Now that we have the circadian clock, we sleep at night, or mostly. So some more information about that. So actually, when the brain decides that we go to sleep, what happens is that neurons shut down here all the arousal systems, and these start from neurons in the hypothalamus. So they send signals to thalamic areas, and then they go over to cortical areas, saying the brain that now you actually go to sleep. And then this connects with all the other organs in our body. We reduce the muscle tone, and we go to sleep. When we need to wake up, then actually it starts from the brainstem that gives information to the forebrain, to the thalamus, and through the thalamus it goes to the cortex, and then this is the signal that says now you can actually activate all the system, that the sensory motor system. Actually, we start sensing, our senses are alert, okay? And this is how we wake up. So these mechanisms, they compete, and we wake up or we go to sleep. On top of that, we have this circadian rhythmicity that I told you about. And to put it in a simple context, so here is the eye, and there is a group of nerves in the retina that they act as light detectors. So these light detectors actually give, they go into the hypothalamus, they enter here, it's the suprachiasmatic nucleus, and when we see light, then there are certain hormones that are secreted, and depending on the time of the day, we go to sleep, or they help to be awake. Okay, so let's assume that our brain decided to go to sleep. And let's see the sleep architecture. So when we talk about sleep architecture, we are talking about the different stages that the brain goes through throughout the night, throughout the sleep cycle. We harshly divide sleep into big stages, the REM and the non-REM. The REM stands for rapid eye movement and non-rapid eye movement. Additionally, non-REM is divided into 1, 2, 3, and 4 stages. So just for you to be able to read this diagram, the blue areas are the non-REM sleep. The magenta is the REM sleep. And here it shows during the night, 8 hours of sleep, how the brain goes through the different stages. So as you see, it's not that the brain enters the non-REM and then goes into REM and then it wakes up. No, it's a cycle. So normally, a normal brain, as soon as it falls asleep, it enters the non-REM sleep. It goes out to REM, then to non-REM, REM again, and this is a cycle. So what does it mean, REM and non-REM? Well, these are two very different brain situations, let's say. So they differ in many physiological processes. I have a table here. You can see they don't differ only in the brain activity, but all the physiological processes in our body differ between REM and non-REM. So non-REM generally is considered to be a stage, and we will give some more information about that later, a stage where we sleep deeply. It's the deep sleep. REM is considered to be a more light sleep. It's the time of the night where we dream. And if we look at the signals, so here we have EEG signals of the brain during wakefulness, and these are the signals during sleep, REM sleep, and 1, 2, 3 stages of non-REM sleep. So what you actually see is that wakefulness resembles REM sleep. And this is actually what we say, that an EEG during REM is very similar to an EEG during wakefulness. And one very interesting fact is that we have muscle atonia during REM sleep, so that we don't actually act during our dreams. On the other hand, during non-REM, which is the deep sleep, brain, what actually does is it calms down, the networks calm down, and it's very different from wakefulness. What we see is very, very different from wakefulness. So the great importance of non-REM sleep, as far as we know until now, at least, is the memory consolidation. So we don't exactly know the mechanisms, but we do know that during non-REM sleep, what brain does is that it consolidates the memories that came during the day. And they are not memories yet, they are experiences. So how an experience becomes a memory, this is decided during the non-REM sleep. We don't know exactly how, but we know when. Okay? So how we actually, sorry, just for to be clear, So traditionally, we divide non-REM sleep in 4 stages. So what you see here, 1, 2, 3, and 4. And 3 and 4 stages are the deepest stages of non-REM sleep. But the newest literature and guidelines just give stage 3. So 3 and 4 are collapsed, and they give stage 3. Because actually, sometimes stage 3 don't even exist, or we cannot even distinguish them, because the way to distinguish them is the EEG. I will show you now. So just for you to be clear, that's why here we only have 3 stages. Here it has 4, and here it has 3. So when we say here 3, we mean 3 and 4, the traditional division. So here you see the EEG in wakefulness, the stage 1 EEG, stage 2, and stage 3, which is considered, as we said, the deepest sleep. You can see that the brain rhythm is very, it's nothing but similar. As the night goes by, and to be more correct, as the cycle of REM non-REM goes by, and we enter deep sleep, what we see in the EEG is high voltage synchronized activity. These waves are only seen during the deepest sleep in stage 3. And here what you see is that we start seeing them as non-REM, as we enter in non-REM, but we see them regularly in the deepest stage of non-REM. So this activity is called slow waves or slow oscillations. It's one type of brain activity, of brain rhythms. The way we divide the brain rhythms is by their frequency. And this is exactly as its name, slow waves. So they have a frequency of 0.5 to 1 to 2 hertz. So why do we actually care about the slow waves, why they are so important? First of all, take a look at this picture here. So wakefulness, non-REM, and REM. And these are raw EEG signals. So you can see low voltage EEG signal during wakefulness, a bit higher voltage here, but very similar to wakefulness. And here, the non-REM EEG signal. So here is depicted the neuronal activity of the cortex. And you can see here that during wakefulness and during REM, the activity is completely desynchronized. Let's say it's random. It's not random, but it's desynchronized. But during non-REM, the activity is synchronized. And I will come back to that a bit later. And this is a huge difference. So the importance of slow waves or slow oscillations, it was a very interesting Nature paper, a recent one, 2019. Of course, there is much more work done before that, that showed that actually the fate of a hippocampus-dependent experience that was made during the day is considered from what happens during non-REM sleep in the deepest stages, from what the slow oscillations do. So let's see it in more detail. This is, so here, this area that you see here This is a slow oscillation. And this part here, it's called up state. And this part here, it's called down state. I know it's the opposite of what you see, but this is only technical. The up state, it's the down part, and the down state is the up part, okay? So this is the slow oscillation. The slow oscillation is up state, down state, up state, down state. And this oscillates with a frequency of about 1 Hertz. So this is the slow oscillation, and this is another type of brain activity, not slow waves, but delta waves. And this is another part. Another type, spindles of slow oscillations. What happens is that the network decides that we keep this memory. The memory is consolidated. On the contrast, when the spindle goes in the delta wave and not the slow wave up state, when it coincides with the delta wave, then it promotes forgetting of that experience. Of course, I know this is a very, very, very complex thing. I just wanted to show you that we have insights right now about how non-REM sleep actually helps in memory consolidation. And it's all about brain rhythms, network oscillations, and how these networks oscillate at the same time or a different time. So please keep non-REM sleep, the deepest stage, slow oscillations during that stage, a very synchronized network activity, and it promotes memory consolidation. Here you can actually see again a trace of slow oscillations. I will not stand a lot on that, but here you can see the up state and down state, up state, down state, up state, and down state. And also here, and this is an LFP recording, which means an electrode in the brain tissue and not on the scalp as the EEG. And here you can see the up and down state, up and down state of the slow oscillation. So actually the up and down states are considered to be the cellular counterpart of slow waves. What does that mean? I told you that slow oscillations is the signal we see in the EEG. The up and down states are actually the slow oscillations that we see in LSP recordings, which are the electrodes in the brain tissue. So this is how we see the slow oscillations in the brain tissue, with up and down states like this. So let's have a better look on what happens during the up and down states of the slow oscillation. Here again, a technicality, the up state is the down part, the down state is the up part. So up, down, up, down, with an oscillatory activity frequency of 1 Hertz. So here you can actually see the neuronal population activity, and you can see that during the up state, we have a very high synchronized neuronal activity, which drops during the down state here. It's back again during the up state, drops, up state, drops, and it goes like this. So what we say simply is that during up states, the cortical networks are synchronized and they produce endogenous activity because they are not stimulated somehow. They just produce activity all together, a synchronized endogenous activity, a cortical activity during the up states, and during the down states, the networks are silenced. The neurons are hyperpolarized and the networks are silenced. And then again, the network is driven into a synchronized activity and this oscillates. And here you can see a single neuron. So this is the network activity, and this is one neuron within the network where you can see that here it's active during the up state, it hyperpolarizes during the down state, active, hyperpolarized, etc. So all these data are in vivo data. In 2000, a research group, Sanchez Vives, who is a great scientist and the leading group in the field, they actually showed how we can record this kind of activity, slow oscillations, up and down states activity, ex vivo, from brain slices. And this was a revolution actually, because all the previous data were from cortex were seen from in vivo experiments, and now we can actually put them in the ex vivo or in vitro frame. So they actually shared the procedure on how we can record this activity from a single brain slice. And here what they showed is that the activity ex vivo resembles very well the in vivo activity. So here you can see the recording of one single neuron that enters the up state, the down state, up state, down state. And this is zoom in on this event, up state event. And this is the extracellular signal, the network signal. Okay, so actually during my PhD, what I did was this ex vivo work, these electrophysiological recordings of the networks to record slow oscillations ex vivo. And I tried to go into mechanisms on how certain mechanisms that I will talk to you about regulate slow oscillations, and in the end, how these mechanisms make the network transit into epileptic seizures, from slow oscillations into seizures. And you might be wondering how and why there is a connection there in normal sleep and epileptic seizures. I will try to show you. So what we did here is, here you can see a brain slice. These are mice slices, and we go into cortical area. We use two electrodes. The one electrode records network activity, the LFP, and the other electrode goes into a single cell, a single neuron in that network, and records the single cell activity, the single neuron activity. So on the top left of each slide, I will have either this icon or this icon. So this is the one method I will show you, recording of up and down state activity here. And in some cases, I will use this icon, which is another method, a NIFIS method, which is a voltage clamp where you can actually record activity, GABAergic activity, which is the currents that come from GABA receptors on a single neuron. So first of all, it's very well known that sleep differs across development. Young children have different needs and also different sleep architecture than adults. And this is a very simplistic view to say. So first of all, we tried to show that in our slices as well. So we used young, adolescent, and adult mice, and we recorded the network activity. And what we actually show here is that the activity differs a lot from young to adult. I won't go into many details, but this is the upstate duration. And you can see that the young differ from the adults a lot. This is also the upstate occurrence, how frequent these upstates are, the young from the adults. Also the downstate duration, which is how long the downstates are, they differ a lot. And please note that the adolescent activity is always in the middle. So it either resembles adults or either resembles youngs. And that was very interesting, which is actually the definition of adolescence, probably. In some aspects, it resembles youngs, and in some other aspects, it resembles adults. So we showed this, and we decided to not keep this group for the next experiments, to actually go on with two discrete, very different age groups. So we go on with the young and adult networks. Well, please take a look at the row traces here and see that the events in the youngs are very nice, long, robust with high activity, while in the adults, it's not the case. And if you take a look at the overall signal, you actually see that the young network is a vivid network that has a high frequency of oscillatory activity, up, down, etc., while the adult has lower frequency. Also, this is an extra metric, the multi-unit activity. Young networks exhibit higher multi-unit activity within the upstate event. So all these evidence show that they are quite different networks, so we should treat them differently. So the first part is the role of GABA A receptors. These are receptors in inhibitory receptors in the brain, fast inhibition, and we put a blocker GABAzine to show what happens in the slow oscillatory activity in the brain when we block these receptors. So first of all, just take a look, because I want to be on time, just take a look here on these graphs here. So this is the normal activity in the young networks, and this is the normal activity in adult networks. When we block GABA A receptors, please see here in the down graphs that the normal activity is diminished in young networks. You can see here the red, while in the adult networks is maintained. And what comes up is another type of activity, which are the epileptic events. When we block these receptors, we diminish the normal slow oscillatory activity, and we come up with epileptic activity, and this is the level of epileptic activity in young networks. Please see that the adults also show some epileptic activity, but there is a balance between normal and epileptic events. But in youngs, as soon as we block these receptors, the network is actually messed up. We have seizures. So that was very interesting, and we tried to understand where are these receptors placed? Are they in the synapse or are they extra synaptic? And actually, we show here that these are only synaptic receptors. So when we put our blocker, gabazine, we block the phasic receptors, the GABA receptors that are in the synapse. So here are measures of these currents, of these phasic currents through the synaptic receptors that are blocked. And we have evidence here, because these are the metrics that are affected, that these receptors are placed post-synaptically, in the synapse post-synaptically. So there is something different between the young and the adult networks in the post-synaptic site. And here we quantify this current in young and adult networks. And in the end, we show, I don't want to keep you here. In the end, using diazepam, which is a drug that binds in GABA receptors, we show that alpha-1 GABA receptors are the receptors very important in the young networks, while non-alpha-1 GABA receptors are important in the adult networks. And this is exactly what we have here. So this is the graphical abstract of the paper that's published, that in young networks, alpha-1 GABA receptors are very important for the network to decide to go into an up state from the down state where the network is silenced, to decide to be activated and go into an up state, normal oscillatory activity, or into an epileptic event, which is abnormal, while in adult networks, probably alpha-1 receptors, for some reason, they don't have the same role, or they act differently. So alpha-1 GABA receptors are the key regulators of what the network does when it leaves the down state, and which state to enter, normal or abnormal. Second, it's another model, a knockout mouse, again, the same methodology, but now it's a knockout mouse on alpha-5 nicotinic receptors. And actually, this mouse model is a nice model for studying dendritic inhibition. What's the role of dendritic inhibition on up and down states activity, and how this transits into epilepsy. So this is a nicotinic receptor. I showed you what happens with a GABAergic receptor. Now it's another kind of receptor, nicotinic. So these mice are lacking the alpha-5 nicotinic receptor, and we quantified their pyramidal cells. They seem to be the same. There are no changes in the properties of pyramidal cells, but there is a change in the network activity. So what happens in these knockout mice is that the up and down state activity, the slow oscillatory activity is changed. And just take a look at the raw traces, you immediately see that in the knockout mice, the network is sluggish. And sorry, I forgot to mention that these are young mice. We are talking about young networks only here. So the knockout mice exhibit a sluggish network activity compared to the wild type. Smaller up states and long down states. I won't stand here, just we also, well, we recorded again here the currents, the GABAergic currents on the neurons, and we actually see that probably these neurons in the knockout mice, the pyramidal neurons, receive increased GABAergic input. So receive increased GABAergic input, and we decide to actually, I'm sorry, here, what happened? We add the same blocker that we added before, and we did the young network epileptic, and we add the same blocker here. And what happens in the knockout network is that we don't have epileptic seizures. It's the same age. We would expect to actually the normal activity to drop and for epileptic events to show up. But in the knockout mice, they don't act in that way. So we block the GABA receptors, we expect seizures, and we don't see seizures. So we do see some seizures, but they are much less than in the wild types. So here we have a case where actually we kind of prevent the networks of getting into epileptic seizures through nicotinic receptors. So if we take a look at the circuit of our model, here it's a very simplistic model. Here, what I show you, of course, it's much more complex. So our receptor, the one that we knocked out, is expressed from a certain type of interneurons. These interneurons inhibit other interneurons, SST and PV, which actually inhibit the pyramidal cells that we record from, and they bind in different regions of the cell. And these interneurons bind the dendrites, these interneurons bind on the soma. And so the main source of inhibition from these cells that are knocked out here go into the interneurons that go on dendritic inhibition. So that's how we have a disinhibitory circuit here in our knockout model, which is a very nice model of dendritic inhibition. And what we show here is that if you actually play with dendritic inhibition, you can modulate both the slow oscillatory activity, but also the transition into epilepsy. So the overview of the work was that, first of all, alpha-1 GABA receptors are very crucial for maintaining the endogenous activity, very important for the slow oscillations, and when we block them, we go into epilepsy. And second, the alpha-5 nicotinic receptors, they participate in the circuit, this inhibitory circuit of dendritic inhibition. They, of course, balance endogenous cortical activity, and when we actually, in this model, we block the alpha-1, the GABA receptors, we prevent from going into epileptic seizures. So I hope I gave you some evidence here and tried to somehow convince you that mechanisms that regulate the slow oscillation are also very important for the brain circuits to decide, not decide, but to go into epileptic seizures or to stay in healthy states. So just to close this presentation, here there is a very nice work where this activity, it seems to be a very complex image, but it's not. Just try to follow me a bit. So this is the normal up and down state activity. So this is the slow oscillation here, and this here is the network that enters epileptic activity. This is real-time recording, so you actually see the whole transition of the signal of the network activity from healthy slow oscillation activity into spike wave activity, which this is how we call the epileptic activity here. So you can see that it transits from the one state into the other state. And there are many more evidence coming up the last 15 years and some more earlier than that, that show actually a relation between the slow waves, the slow oscillation activity, and the mechanisms that transit the brain networks into epilepsy. So if we try to understand the mechanisms during slow oscillations, we might also gain some insights into the seizures, the mechanisms that drive the brain networks into seizures. There is a huge work here, and it's very interesting for diagnosis, but also as therapeutic targets there, because maybe a patient, epileptic patient, we could record him in EEG during sleep, and we might see there some biomarkers in EEG and see that, you know, this patient will go into a seizure, and we might be able to prevent this seizure in the future. Yeah, and that was all for me. Thank you very much for your attention. I hope I tried to keep it very simple.
Thank you, Annie. Yeah, you tried, and it was very nice. It was very simple for us, for me, which I'm not an expert in this field. Very insightful presentation. Thank you for your time, for your participation. So I would like to ask if there are any questions, because I have a couple of questions.
Yeah, I have some curiosities. So the first one is, what happens when you break the circadian rhythm? Meaning, if you sleep, like you are used to sleep for 8 hours, and after 4 hours you stop your sleep. What's happening inside the neuronal network?
Yeah, that's thank you for the question. That's very nice. So actually, if you go deeper into this image, you actually see that throughout the night, the REM stage goes more and more and more throughout the cycles. So the percentage of REM is more throughout the night, and it's the opposite for non-REM. So in the beginning of sleep, which is around 4 hours that you already said, you actually reach non-REM. As you sleep, the cycles reach deep sleep. But after that, there is a high chance that in the next cycle, you don't reach non-REM. So as you can see here, so that it goes up. So yes, it matters if you sleep 2 hours or 4 hours or 8 hours, it matters. And yeah.
And this rhythm is affected by, for example, the latitude of inhabitants of the Earth. So, for example, are there any studies about differences in the circadian rhythms between people who lives in the pole or in the equator, because we have a different like night-day shift?
Daylight.
Yeah.
Okay.
Most probably there are huge differences in the circadian rhythmicity because yes, as I showed you, there is a very specific mechanism that, you know, the circadian clock at least starts, it's activated, and it's through the retina, which acts as a light inspector there. So yes, if you are in a country that you live like 10 hours of darkness or the opposite, then it's much different. I'm not aware of specific literature, but yeah, I guess it should be very different, yes.
And are there, sorry.
Just to say, but still you sleep. So even without, if you delete the circadian clock, you still go to sleep. But then it's much more random, and there are other compensatory mechanisms on putting you into wakefulness or into sleep. On the contrast, if you delete the other mechanisms that I talked to you about that regulate wakefulness and sleep, if you delete them, then you have insomnias. Then you cannot go to sleep.
Okay, thank you. And just one more thing. So you have like shown results about the brain of mice, and so do you expect some differences when subjects are humans or like more complex being?
Yes, for sure. There are differences. There are huge differences, of course. But still, we have great insights because, you know, they are mammals. They do have cortex, much more different, but they do have cortex. The brain has a cortex, and of course, networks. And we can get some great insights, even for the human side.
Okay, thank you very much.
You're welcome. Thank you very much.
I would like to ask, based on the first question of Marco and this diagram that you are showing to us, is it the ability to memorize during our sleep, is it connected with the duration of a non-REM sleep?
Yes. So actually, this is a very nice question because so when we are talking about insomnias, there are different disturbances in sleep. So one disturbance might be what we call insomnia, could be the number of cycles, so during the night. The other one would be the length of the cycles. The other would be the percentage of REM and the percentage of non-REM. So there is a huge variability on what is the problem there every time. So yes, as we said, because non-REM sleep is the memory consolidation state of the brain during sleep, when this is disturbed, of course, there are disturbances in the memory consolidation procedure for the brain. And there is a lot of literature coming up right now showing that patients with epilepsies have problems in consolidating memories. And there is another evidence there and the link between the two.
Okay, thank you. Are there any other questions? Okay, let me ask you one more thing. So you've showed to us that blocking the fast GABA mediated inhibition turns the network epileptic. But is it the same with blocking the slow inhibition?
So yes, actually, I showed that GABA receptors are fast receptors. They mediate fast currents, and this is in favor of turning the network epileptic very fast. And we have another type of receptors, GABA receptors, which mediate slower currents. And no, I didn't show you these results. We have them there. When you block these receptors, you don't go into epilepsy at all. You do modify the network activity, but you don't go into epilepsy.
Okay. And last one. So can we learn more about the seizure mechanisms by studying the sleep?
Yes. So that was actually what I tried to at least not convince you, but give you evidence for through this presentation, that the relation of sleep and epilepsy is there, and studying sleep gains there some insights there for epilepsy and the other way around, because there are many similarities in the mechanisms that regulate the balance between the two.
Okay, thank you, Annie, very much.
Thank you.
If there are no more questions, we can